Welcome to the Flowers Lab. In this lab, we're shifting our focus to one of the most specialized and evolutionarily successful plant structures, the flower. So far in this course, you've examined vegetative structures like roots, stems, and leaves. Today, we move into reproductive biology and explore how angiosperms reproduce, diversify, and dominate terrestrial ecosystems. Flowers are not just decorative structures. They are highly modified shoots designed for sexual reproduction. And like everything else we've studied in this course, every floral structure exists because it serves a function. By the end of this lab, you should feel confident identifying the major parts of a flower, understanding how pollination and fertilization occur, and recognizing how floral variation reflects evolutionary strategies. Before we begin, let's take a moment to review the learning goals for this lab. By the end of this activity, you should be able to describe the four major floral whorls and explain what each one does. You should be able to distinguish between superior and inferior ovaries, which will become very important when we start dissecting specimens. You'll also learn the difference between complete and incomplete flowers and perfect versus imperfect flowers. Those terms sound similar, but they describe very different things. We'll walk through the angiosperm life cycle and connect flower structure to double fertilization, one of the defining characteristics of flowering plants. And finally, you'll begin to recognize evolutionary trends in flowers, including symmetry, fusion, reduction of parts, and pollination strategies. As always, our guiding theme remains the same. Structure supports function, and in flowers, structure supports reproduction. Flowering plants, angiosperms, make up one of the largest and most diverse groups of plants on Earth. Their defining feature is the flower. Flowers are specialized reproductive shoots. They are not separate from the rest of the plant body. They are modified stems with modified leaves arranged in whorls. What makes angiosperms especially successful is their reproductive innovation, including enclosed ovules and the process of double fertilization. Flowers also drive co-evolution. The color, shape, scent, and timing of flowers often match specific pollinators. Bees, birds, moths, bats, each one has influenced floral evolution. So when we study flowers, we are studying reproduction, ecology, evolution, and plant diversity all at once. Most flowers are built from four basic whorls, layers of modified leaves arranged concentrically. Starting from the outside, the calyx, made of sepals, protects the developing bud. The corolla, made of petals, usually attracts pollinators. Together, the calyx and corolla form what we call the perianth. Next is the androecium, the male reproductive layer made of stamens. Each stamen consists of a filament and an anther that produces pollen. Finally, at the center is the gynoecium, composed of carpels. Carpels are modified leaves that enclose ovules. Collectively, carpels form the pistil, which includes the stigma, style, and ovary. All of these structures are attached to the receptacle, which sits at the tip of the flower stalk or pedicel. As we dissect flowers today, I want you to think of these parts as modified leaves arranged in a highly organized reproductive system. Let's focus more closely on the reproductive structures. The stamen is the male structure. It consists of a filament that supports the anther. Inside the anther, pollen grains are produced. Each pollen grain carries the male gametophyte. The pistil is the female structure. At the top is the stigma, which receives pollen. The style supports the stigma and provides a pathway for pollen tube growth. At the base is the ovary, which contains ovules. Inside the ovary are chambers called locules, and ovules are attached to the ovary wall at the placenta. Remember, ovules become seeds after fertilization. This is where reproduction actually happens. A complete flower contains all four floral whorls: sepals, petals, stamens, and carpels. That means it has both protective structures, attraction structures, and both male and female reproductive parts present structurally, whether or not they are functional. This spiderwort is a complete flower. An incomplete flower is missing one or more of these whorls. Maybe it lacks petals, like the powder puff tree. Those puff flower heads are actually masses of stamens. Maybe it lacks sepals. The American elm flower is missing sepals and petals. Maybe it lacks stamens or carpels. This eastern cottonwood has separate male and female flowers. This classification is purely structural. We're asking, are all four whorls present? This is different from asking whether both reproductive sexes are present. And that leads us to the next distinction. Again, think structure first. Count the whorls. Don't overcomplicate it. Now let's shift to perfect versus imperfect. This classification is about reproductive organs only. A perfect flower contains both stamens and carpels. It has both male and female reproductive structures in the same flower, like this ardesia flower. An imperfect flower contains only one sex. If it only has stamens, it's a staminate flower. If it only has carpels, it's a carpellate flower, like on this hazelnut tree. Here's where students often get confused. A flower can be incomplete, but still perfect. For example, it might lack petals, but still contain both stamens and carpels. So remember, complete versus incomplete equals structural whorls. Perfect versus imperfect equals reproductive sex. Two separate systems. When you're dissecting flowers in this lab, I want you to mentally separate those questions. Now that we've talked about perfect and imperfect flowers, we need to zoom out one level and talk about the whole plant. This is where monoecious and dioecious come in. A monoecious plant produces separate male and female flowers, but they occur on the same individual plant. So one plant has staminate flowers and carpellate flowers, just in different locations. Corn is a classic example. The tassels at the top are male, the ears are female. Same plant, separate flowers. Now contrast this with a dioecious plant. In dioecious species, male and female flowers occur on completely separate individuals, like in this American holly. That means one plant is male, another plant is female. If you only have male plants, you will never get seeds. If you only have female plants, you will also never get seeds. This has major implications for ecology, agriculture, and horticulture. From a biological standpoint, dioecy increases the likelihood of cross-pollination and genetic diversity. Monoecious allows flexibility but still separates the sexes within a single organism. So let's connect this to earlier terms. Perfect versus imperfect describes individual flowers. Monoecious versus dioecious describes the entire plant. Different levels of organization, and once again, structure supports reproductive strategy. Now let's look at pollination strategy. This is where structure becomes incredibly obvious. Animal-pollinated structures are designed to attract something. They often have bright petals, they may produce fragrance, they usually offer nectar as a reward. Their pollen is often sticky or textured so it adheres to pollinators. Everything about these flowers is intentional. Color, scent, timing of opening, all tuned to a specific animal. Now contrast that with wind-pollinated flowers. Wind doesn't care about color. Wind doesn't respond to scent. Wind doesn't need nectar. So wind-pollinated flowers often have very reduced or absent petals. They produce massive quantities of lightweight pollen. Their stamens are often exposed. Their stigmas are often large and feathery to catch airborne pollen. Again, structure supports function. If pollination depends on precise delivery by animals, the flower invests in attraction. If pollination depends on probability and air currents, the flower invests in quantity. When you start recognizing these patterns, you can often predict the pollination strategy just by looking at floral structure. Pollination occurs when pollen lands on a compatible stigma. Each pollen grain germinates and grows a pollen tube down through the style to reach the ovule. Each pollen grain carries two sperm cells. One fertilizes the egg, forming a zygote. The other fuses with the central cell to form triploid endosperm, which nourishes the developing embryo. This process, called double fertilization, is unique to angiosperms. It's one of the major evolutionary innovations that contributed to their success. Before we jump into the first activity, let's talk about what you'll actually be doing today. You'll see real flower specimens that are dissected and identify the 4 floral whorls. You'll compare monocot and eudicot flowers and begin recognizing patterns in floral organization, including number of parts and symmetry. You'll determine whether ovaries are superior or inferior and what that tells you about flower structure. You'll also move to microscopic images to examine ovule development and pollen development, connecting structure to the life cycle of angiosperms. And finally, you'll apply what you've learned to a case study on pollination, where you'll analyze how flower traits reflect specific pollinators. Just like in your other labs, you'll be making detailed labeled drawings in your notebook. Focus on accuracy of structure rather than artistic detail. By the end of this lab, you won't just be able to name flower parts, you'll understand how those parts work together to ensure reproductive success.